All right, so chapter 9 is going to focus on respiration and fermentation. Both are processes that occur in our cells that help us generate energy that can be used to allow the cell to do its various work activities. Cells need to get their energy from external sources. Autotrophs are able to get their energy by creating it through photosynthesis, and heterotrophs will get their energy by either eating organisms that are able to photosynthesize plants or by by using other organisms as their food sources that do eat plants. And energy is going to move into our ecosystems through sunlight. It's going to leave as heat. Again, photosynthesis is one way that we can generate those organic molecules that we can use as chemical energy. It also is able to provide us with sources of oxygen, which is super helpful to be able to get lots of ATP molecules from those organic substances. And then respiration is able to break those down to create ATPs, to reform those ATPs, which then can be used to do work. So again, we're going to spend a little bit more time with photosynthesis in chapter 10. We're going to be focusing on the part at the bottom right now, a respiration, which takes place in the mitochondria. Notice the folds, the cristae. So that's going to play a pretty important part in cellular respiration, as is the matrix, which is what is found inside of that inner membrane. And so we see here, this is another one of those processes where they're kind of paired together, how respiration is able to generate carbon dioxide and water, which then can be used in plants as photosynthesis, whether they make it directly or whether they obtain it just by being outside and getting those resources through water and through the air. And then they are able to create those organic molecules as well as oxygen, which are then used again in respiration. So exergonic and endergonic processes paired together. So catabolic pathways are ways that we are able to get energy by oxidizing those organic materials. Fermentation is able to oxidize those organic molecules, but not to the extent that we see with respiration in terms of generating ATP. It does it without oxygen. And respiration as a whole is both aerobic and anaerobic. We're going to be talking about aerobic respiration, which is what our cells do primarily. And then fermentation would occur in your anaerobes, those that do not need oxygen, or at times in our cells when we don't have enough oxygen to be able to use aerobic respiration as our primary energy source. We don't have to just be looking at sugar molecules as a means to trace respiration. Any of the macromolecules that are consumed as fuel, whether it be proteins or fats, they too can go into the cellular respiration process. The redox reactions, oxidation reduction. Electrons can be transferred in chemical reactions. Those electrons are stored in your organic molecules, and when they're transferred, they can release energy. That energy can then be used to make ATP. You must have something undergoing oxidation in your reaction as well as something undergoing reduction. That's why they're called redox. You can't have just one, you have to have both. In oxidation, you tend to have in oxidation, you do have electrons being lost. You have the positive charge of the substance undergoing oxidation increasing, while reduction, the exact opposite takes place. Electrons are being gained and the positive charge is being decreased. And so there's just a classic example between sodium and chlorine, why they make the ions they do. Sodium loses an electron, chlorine gains its electron. But when we're going to be talking about cellular respiration, both aerobic and anaerobic, we're not going to be dealing with metals and nonmetals necessarily, which is the more classic ion set form. I will say a little bit more about that before we get in, a little bit more about redox before we get into that. The substance that's undergoing oxidation is giving up electrons. It's acting as an electron donor. So it itself is known as the reducing agent. It provides a way for reduction to occur by being a source of electrons that then can be gained. The oxidizing agent is a substance undergoing reduction. It gives a place for the electrons to be lost to. So I know that seems kind of weird that the substance undergoing oxidation, the element that's present in there that's undergoing oxidation, the substance that houses it is the reducing agent and vice versa. But again, the agent facilitates the opposite process. What we're going to see with cellular respiration is that we don't physically have a transfer of electrons from our organic molecules to oxygen. But what we do have is a difference in how those electrons are being shared in the covalent bonds. So here we have glucose and it's going to CO2, and we have oxygen and it's going to go to water. So the carbon that's a part of glucose is bonded to a lot of hydrogens. And whereas in CO2, it is just bonded to oxygen. Oxygen is much more electronegative, and so it's going to pull the electrons in its bond towards oxygen as opposed to towards carbon, which is why the carbon is able to undergo oxidation. When it's paired with hydrogen, it's pretty much paired with something that's the same in terms of electronegativity. It really doesn't have any real difference in the sharing of the electrons. But when you get with oxygen, because of its electronegativity, the electrons get pulled towards it, and so carbon technically is losing electrons. When oxygen is by itself, it's neutral, and when it's part of a compound, for your purposes, there's a little bit more to it that we get into with AP Chem, it's going to have a -2 charge. And so we're going to be assigned 2 of the electrons. So oxygen is decreasing in oxidation number. It's becoming less positive, and so that's why oxygen is being reduced. So in cellular respiration, we basically are breaking down glucose or whatever our organic molecules are that are our energy sources, but we don't break it down all at once. We break it down through a series of steps, and by doing that, we're able to harness the electrons that we pull as we break apart that glucose molecule into smaller and smaller pieces. And so we're basically decoupling oxidation from reduction. We have to have some electron carriers to be able to do this, to separate those two processes, because we need the electrons to end up with oxygen because oxygen needs to be reduced. It needs to gain those electrons. So there are two helpers that make this possible. NAD plus is one of them, sometimes called NAD. And these helpers, in NAD's case is a coenzyme, are able to have both oxidized and reduced forms. And so when they accept an electron, they also accept a proton. And because they're accepting an electron, they're being reduced. And so NAD plus is able to act as an oxidizing agent. When it is in NADH form, the reduced form, it has energy now that we can use to make ATP. So the less energetic form is its oxidized form. Again, as you pull those hydrogens and the protons and the electrons out of your organic materials, dehydrogenase is able to allow it to convert to the reduced form of NADH, and it'll also help that NADH form be oxidized back into NAD+. So NAD, and there's another electron carrier, FADH2, that can help to move these electrons through the electron transport chain, and we'll see them happen in a series of steps. These electron carriers in the electron transport chain are found in the inner membrane of the mitochondria, but for prokaryotes that don't have organelles, they're found in the plasma membrane. Oxygen is at the end of these at the end of the proteins that are present in the electron transport chain. And it basically, because it's so electronegative, helps to pull those electrons towards it, and eventually it'll form water and provide a means of reforming ATPs. So if we just did it at once, you'd have a lot of heat, a lot of light energy release, but it'd be really hard to harness it. By breaking it into smaller and smaller chunks and grabbing onto each of those at a time, you're still not going to be able to absorb all of the energy that was present in your fuel source, but you're going to get a lot more bang for your buck than you would if it happened all at once. So within cellular respiration, there are three processes: glycolysis, the citric acid cycle, and oxidative phosphorylation. So glycolysis is the first step where we take glucose, which is a 6-carbon molecule, and we break it into two 3-carbon molecules of pyruvate. Citric acid cycle is where we take the pyruvate and we finish breaking down the glucose. We do have an oxidation process to pyruvate prior to it entering the citric acid cycle to be able to make that happen. And that's the part that involves the oxidation of those two steps are what are going to be involved in oxidizing glucose. The oxidative phosphorylation is where we're going to end up making our ATP and reducing our oxygen. Now, not every cell, prokaryotes specifically, but not everybody is going to be able to enter the citric acid cycle or go through the electron transport chain as well as chemiosmosis, oxidative phosphorylation. If oxygen is not going to be available, or if you don't have the mitochondria organelles present, the only part of cellular respiration cells are able to undergo is glycolysis. Both glycolysis and the citric acid cycle are able to make some ATP, and they are able to make it using enzymes. It's substrate level phosphorylation, where an phosphorylated intermediate transfers its phosphate group to ADP, and that is how ATP gets made. Oxidative phosphorylation is going to come about because of the reduction of that oxygen molecule. There are at least one other type of phosphorylation, but those are the ones that we're going to be dealing with in this unit. So glycolysis, again, is taking place in your cytoplasm. Electrons are pulled out of the glucose molecule as it's broken down via NADH. The pyruvate is oxidized a little bit further so it can enter into the citric acid cycle. It too is going to make some NADH there as well as FADH2, and it's going to generate some ATP. And all of those electrons that have been harnessed in your carrier molecules, the NADH and the FADH2, will then subsequently end up along the electron transport chain, and we will make ATPs as well as water. So oxidative phosphorylation, although it's only present in one step, is what makes the majority of our ATP in cellular respiration by taking the electrons from the decoupling of oxidation and transferring them to oxygen so that the oxygen molecule can be reduced. There are a few molecules, as I described, that are made using substrate level phosphorylation, both in glycolysis and the citric acid cycle. Those are shown to you that what's happening there below is an example of substrate level phosphorylation. But generally speaking, every glucose molecule that is broken down into carbon dioxide and water through respiration can make approximately 32 molecules of ATP. It is not an absolute number, and we'll talk about why as we continue through this. So section 2 is just going to focus on glycolysis, how we can break down our organic material into pyruvate molecules. Again, glycolysis, splitting of sugar, is taking that 6-carbon molecule and breaking it into the 2 3-carbon molecules, and it's also generating 2 ATPs. There are 2 phases to it, both an investment and a payoff. So for glycolysis to take place, 2 ATPs have to be consumed to help to break apart the glucose molecule into pyruvate. But as a result of that process, 4 ATPs get made, so there's a net increase of 2 ATPs. And 2 of the NAD plus molecules, those oxidized electron carriers, are able to pick up electrons to form their reduced form to NADH. And then those pyruvate molecules are able to continue on either aerobically or anaerobically with respiration. Again, you do not have to memorize all of these structures, just to give you a little bit better understanding as to what actually is taking place in both the investment and the payoff. What I need you to be familiar with is what is being made and what happens to glucose as it goes through these series of steps. All of those steps were catalyzed by enzymes. The citric acid cycle is going to finish off the oxidation of organic molecules, but there is a kind of a pre-step that has to take place before you can enter into the citric acid cycle. There does have to be oxygen present. There do have to be mitochondria organelles present for this to occur. We'll talk about what happens if neither of those or if one of those is not present at the end of this presentation. But we have to prep our pyruvate molecules and turn them into acetyl-CoA. So we need to oxidize the pyruvate a little bit further. We need to remove an additional carbon. And we're going to see that this takes place. It's going to involve three major steps to make that acetyl-CoA acetyl coenzyme A. So in the cytoplasm, before the pyruvate can enter into the mitochondria through a transport protein, it's got to have one of its carbon dioxide groups removed. We need to oxidize another one, sorry, we need to reduce another one of those carriers. We need to turn NAD plus into NADH, and then we need to join coenzyme A on to what is left of the pyruvate molecule. So keep in mind this has to happen for each pyruvate molecule. So for one glucose molecule, at this step, you would have two carbon dioxide molecules given off, two NADH electron carriers formed, and two acetyl-CoA molecules that are ready to go now into the citric acid cycle. So in the citric acid cycle, which is named also, or also called the Krebs cycle for the scientist who first described it, we finish off breaking down pyruvate into CO2. Because we still have more carbons to pull off, each time we go through the citric acid cycle with one of those acetyl-CoAs that we formed from pyruvate, one ATP, three NADH, and one FADH2, those electron carrier molecules are formed for each term. Um, there are a total of 8 steps with specific enzymes for what happens at each step. I don't expect you to know what those different steps are, but I do want you to realize that when acetyl-CoA becomes a part of the cycle, it combines with an organic molecule that is present, and that organic molecule is reformed as the acetyl-CoA goes through that cycle so that it's ready for another round. And then we are able to have the NADH and the FADH2 pick up electrons, which can be moved subsequently from the continuous breakdown of the pyruvate molecule to the electron transport chain. So there's your pyruvate. And again, each time it comes in, you're going to go through the whole cycle, but one glucose molecule is going to go through this twice. So if we have one glucose molecule after the pyruvate has been oxidized and exited from the citric acid cycle, you will have made not 4 NADHs, but 8. You will not have made 3 CO2s, but 6. You will not have made 1 ATP, you'll have made 2. And you'll have not made 1 FADH2, you will have made 2 of those as well. So everything gets doubled. So there's a fun, separate, all the different steps in the citric acid cycle that I had to memorize in biochem. No, no, no. Big picture. What's coming in, what's going out. Oh, sorry, I didn't mean to put that question in there. What happens to each of the carbons in glucose as a result of glycolysis, pyruvate oxidation, citric acid cycle? So glucose is going to split it. Pyruvate, you're going to lose one of your carbons when you go through oxidation. So now you have two carbon molecules. And as you continue through the citric acid cycle, the rest of the carbons that were bonded initially in the glucose molecule are separated off and are given off as carbon dioxide. Okay, I guess I did plan to have that one in there. Sorry, forgot. So there's your glucose. Glycolysis splits into pyruvate, oxidation splits it into the two carbons, and then citric acid cycle makes the rest of your CO2s. So now you have one of your two products from respiration. Now let's see how we get our other product and how we get our ATP. So oxidative phosphorylation, chemiosmosis is going to take the electron transport chain and couple it to the ATP synthesis. So you have all of these NADH carriers and all these FADH2 carriers that have the electrons from the step-by-step breakdown of your glucose molecule, and they are going to transfer these electrons to oxygen. And as they do that, those carriers are going to get oxidized, and the oxygen is going to be able to form water. And we're going to see how that's able to power ATP. So the electron transport chain again is on that inner membrane, that folded area. That's part of the reason the mitochondria inner membrane is so folded. It increases its surface area. Lots of places for this electron transport chain to take place. There are several proteins that make up the electron transport chain, and the electrons get passed through these. They're moving from cytochromes to oxygen. As they go through them, those the proteins are going to have less free energy available, and they are going to become more electronegative, going down to your final electron acceptor, which would be oxygen. When the electrons are transferred, they are going to again turn your NADH and your FADH2 back into their oxidized forms. So all the electron transport chain is doing is moving those electrons along to finally get to oxygen. So, and by doing it in smaller chunks, it makes it a little bit easier to manage the energy transfer that's taking place. So there you can kind of see what we're talking about with the cytochromes and the irons, how the electrons are moving from protein 1 to Q to protein 2 to protein 3 to cytochrome C to protein 4, and then finally to oxygen. But as they move through those multi-protein complexes, the free energy compared to oxygen is decreasing, and oxygen, being your terminal electron acceptor, is helping to pull those electrons through. Why we need to have these electrons pull through, because we still haven't made any ATP. All we've done with that so far is make water. Well, when the electrons are transferred, they are able to, the energy that's released is able to help move hydrogens from the mitochondrial matrix to the space between the two membranes, the inner membrane and the outer membrane. So active transport, they're able to help push them through. But the hydrogen doesn't want to stay out there. It's going to diffuse along its concentration gradient back through the membrane. But there's only one place where the hydrogen ions, their charged particles, are able to move back through the cell membrane, and that is through a special protein, an enzyme, ATP synthase. And you're going to see how ATP synthase is able to use the flow of those hydrogen ions to make ATP. And so this would be an example of chemiosmosis. So we're taking the redox reactions that are occurring along the electron transport chain, and we are using the energy that is stored in that hydrogen gradient to make our ATP. And that hydrogen gradient often is called a proton motive force, since we're able to use that hydrogen gradient to do work to make those ATP energy molecules that then can be used throughout the rest of the cell. So this is probably the wildest protein I've ever seen. It basically is like a knob, and you can see how those hydrogen ions, basically, again, they can only come in through this channel. They have to fill in to those individual spaces, and as they fill into those individual spaces, they turn the knob, and as the knob is turned, and that energy that is generated can then be used to put another phosphate group on ADP and make ATP in the mitochondrial matrix. And as they move the rotor, the different hydrogen ions will move back into that mitochondrial matrix, and additional hydrogen ions will come back in from the inner membrane space and take spots along that rotor. So the electrons are moving through. Those would be your yellow arrows. As the electrons move through, they're able to push hydrogens out into that inner membrane space. We're able to reduce our carriers. The hydrogens come back in, they diffuse along that channel, the ATP synthase, and because that is the enzyme that catalyzes ADP and phosphate to make ATP, the energy that is generated by the hydrogen ions coming back in is able to be then used to make your ATP molecules. So this whole process is oxidative phosphorylation. The first part, obviously, where water is made is the electron transport chain. The second part, where ATP is made using the hydrogen gradient, is your chemiosmosis. So we've talked about ATP being made in a lot of different places. The flow of the energy is going to be the same with aerobic respiration. You're going to take your glucose molecule, you're going to start to break it down into NADH as well as FADH2. That's going to move into the electron transport chain. Then we're going to have the energy get transferred to that proton motive force, which then can be used to make ATP. If everything went perfectly, every NADH could make 3 ATPs at a maximum, and FADH2s, each of those can make 2 ATPs. But we do not get 100% efficiency. Only about 34% of the energy that's present in a glucose molecule actually gets transferred. But as I talked about in class, when you are driving, you only get to consume about 10% of the energy in gasoline. So this is still pretty efficient, all things considered. It's not an absolute. You are going to lose some energy in the form of heat, but this is more of a theoretical value. So just overall, you've got your glucose to pyruvate, your pyruvate to acetyl-CoAs. They go through the citric acid cycle. All the energy, the electron shuttles, the NADHs, the FADH2s are going to help to move those electrons into the inner membrane of the mitochondria, and then oxidative phosphorylation will be able to take place. Not all of those 32 ATPs I talked about are going to be formed through oxidative phosphorylation. You get a couple made in glycolysis and a couple made through the citric acid cycle. And you can see where the reason again we talked about with why you have twice the number of NADH carriers and twice the number of FADH2 carriers coming out of pyruvate oxidation and the citric acid cycle is because once that glucose molecule is split in glycolysis, each of the subsequent pyruvate molecules and then acetyl-CoAs have to go through the citric acid cycle. So we spent a lot of time talking about aerobic respiration. We're going to talk now about anaerobic respiration. We've talked about how you have to have oxygen to be able to go through the citric acid cycle and the electron transport chain. You don't really use oxygen in the citric acid cycle, but there's no real need to go into the citric acid cycle if you're not going to be able to do anything more with those carriers. You also aren't going to be able to go through the citric acid cycle or the electron transport chain if you don't have mitochondria. So with that in mind, glycolysis, rather than making a half-hearted effort to do citric acid if it's just that you don't have oxygen present, is going to be able to use fermentation, which is a form of anaerobic respiration to make ATP. It's not nearly as effective as what you would get if you were using oxidative phosphorylation, but it does get more ATPs for you. So the electron transport chain in an anaerobic respiration is not oxygen, and we'll see what it is instead, something less electronegative. And when this occurs, instead of using oxidative phosphorylation, it uses substrate level phosphorylation to make your ATPs. So there's lots and lots of different types of fermentation. We're going to focus on alcoholic and lactic acid fermentation. Part of fermentation is going to result in reforming our electron shuttle, taking the reduced form NADH and using it to regenerate the oxidized form NAD+, so that those electron shuttles can be reused in glycolysis. So alcoholic fermentation is going to require pyruvate to be converted to ethanol. In order for that to happen, it's got to lose another one of its carbon molecules. It'll do that with CO2, and it is what is used to for yeast. It's used in making wine. It's used for baking. Lactic acid fermentation is what you most likely have experienced with sore muscles. And you do not have to pull off another carbon from the pyruvate molecule. You do end up making lactate by some rearrangements. You will generate that NADH molecule again, and you'll also make some ATP. This is also a way in which cheese and yogurt can be made. So that's just a summary of what's happening in glycolysis and how that works with fermentation. You have the 2 ATP that get made in glycolysis. You make your pyruvate. Again, in alcoholic fermentation, the pyruvate has to have a carbon dioxide molecule removed, similar to the oxidation and pyruvate of acetyl that turns it into acetyl-CoA. But in this case, it turns it into acetaldehyde, and then the acetaldehyde is reduced further into ethanol and reforms the oxidized electron shuttle, NAD+. While in lactic acid fermentation, the pyruvate molecules go straight through and they form the lactate. You can see how there's just a slight change in the structure between pyruvate and lactate, but that slight change allows the NADH carrier to be oxidized again. So anaerobic versus aerobic. Both of these are going to use glycolysis. They're both going to result in oxidizing your organic molecules and getting energy from those food sources. They're going to make roughly 2 ATPs. Both of these happen to use NAD plus as their oxidizing agent to help to pull off the electrons during glycolysis. But the final electron acceptor is going to differ between the two. We're using a less electronegative electron acceptor with the anaerobic respiration, the pyruvate or the acetaldehyde, while in aerobic you're using oxygen. You do not make any more ATP when you go through fermentation, so the 2 ATPs that you got from glycolysis are all that you get with anaerobic respiration. While with aerobic respiration, much more bang for your buck, approximately 32 ATPs per glucose molecule. So obligate anaerobes, I've talked about these verbs a little bit earlier. They can only do fermentation or anaerobic respiration because they cannot function with oxygen. If they are placed in the presence of oxygen, they will die. Yeast and bacteria are able to use both fermentation or cellular respiration. They're okay without oxygen, they're okay with oxygen. So pyruvate, because you can go into the citric acid cycle and oxidative phosphorylation, has the option to do that if there's oxygen present, or it has the option to go through fermentation if oxygen is not present. All right, and there you can kind of see where that fork in the road is between the two processes. So glycolysis has a lot of evolutionary significance. It's thought that before we had oxygen in our atmosphere, um, ancient prokaryotes were using glycolysis. They had to have some way to form ATP, so it's felt that this is probably one of the more conserved processes that takes place in cells. I talked earlier about how you don't just have to have sugars to go into glycolysis. You can use proteins and carbohydrates and fats, and here you can kind of see where they would fit in. You can take amino acids from proteins and feed them into pyruvate. You can take glycerol and feed it into one of the intermediates before you get to pyruvate, and how you can take fatty acids and feed them into acetyl-CoA, and that you can take those amino acids and feed those also into acetyl-CoA as well as a citric acid cycle. So there's lots of ways to take the macromolecules that that we ingest or that organisms ingest and feed them into this metabolic pathway. So regardless of what is being fed in, things do have to be broken down to a certain point to be able to be consumed in the citric acid cycle even further. We can't just take straight meals and feed them into this pathway. So we have to digest our proteins, we have to digest our fats, and we would have to digest some of our more complex carbohydrates. Some things that are pertinent about this is that if you are going to oxidize a fat molecule as opposed to a carbohydrate molecule, you're going to get a lot more ATP from it. Anabolic pathways are going to be ways that our body is able to build larger substances. We might be able to get some of these from glycolysis or from the citric acid cycle, or they could just come from our food sources. We spent some time with feedback controls in chapter 8, and we're going to see that respiration has lots of places for feedback mechanisms. It's got all those enzymes that are controlling all the different steps, both in glycolysis as well as in the citric acid cycle. So if your ATP concentration is dropping, so your respiration process would speed up because your body knows that you need more ATP to conduct your various activities. And if there's plenty of ATP, then your respiration tends to decrease. So PFK is probably phosphofructokinase is one of the key places that can impact on whether respiration is increased or decreased. If there are large concentrations of ATP present, it will shut that enzyme down and it'll inhibit it so that it won't continue along glycolysis or the rest of the processes that are going to take place. And the same thing would happen with citrate. But if you do not have lots of ATP because you have broken down your ATP and you have AMP present, AMP again would be 1-phosphate, adenosine monophosphate, that would stimulate phosphofructokinase to continue to break down glucose and so that, or whatever your organic material is, so that glycolysis in the citric acid cycle and oxidative phosphorylation can occur. So this is just kind of a summary of glycolysis and the citric acid cycle, the molecules that are going in and the molecules that are coming out. And we've already spent some time. There's no real breaking down of glucose anymore after this step because these are the two parts where oxidation of your organic molecules occur, and the electron transport chain would be where you have the reduction of your oxygen.